Hi everyone, it's Mr. Sinti, and today I'd like to talk to you about an aspect of our immune system that is so crucial that it's really amazing. And the fascinating thing about it right out of the gate is that it involves a type of white blood cell that circulates in our body called a lymphocyte. And these lymphocytes can secrete these Y-shaped proteins that look a lot like lobsters, and I mean that. They look a lot like lobsters, and these proteins circulate through our body. They circulate through our bloodstream and through the lymph and in the tissue fluids, and they help to protect us against a variety of germs. And so that's what this particular video is going to be all about, which is the secretion of antibodies and how they work to effectively destroy extracellular pathogens. And when I say extracellular pathogens, I mean like free, meaning like on the outside. It's a little bulky there. Let me turn that down. So on the outside, free pathogens. So if a pathogen gets inside of your cell, you're going to need a T cell to effectively destroy the cell, and that's called cell-mediated immunity. But this particular discussion is on humoral immunity, which is the secretion of antibodies. And so it's initiated when a B cell, which is a type of white blood cell that's a lymphocyte, it has this very large nucleus, it's a B cell. And when it encounters an antigen, now an antigen could be a microorganism that's foreign to the body. So we have an antigen, and it has these foreign proteins on its cell membrane that the T cell is going to recognize as being foreign. And it's going to recognize it because there's specific receptor proteins that are located on B cells that are specific to a particular germ. And so what's remarkable is, now it's difficult to illustrate this because I would have to draw in hundreds of thousands, but I'll just try to do this a little bit. There's an amazing potpourri of different kinds of B cells, and they're all similar in a sense that they all can produce antibodies, but their surface proteins vary. And their surface proteins, I'll just sort of make this one V and this one sort of like little U's. I'll make this one W, like that. I'm losing creativity here. Like this, and then maybe something like wacky like this. So the surface proteins mimic the particular type of antibody that the protein is going to secrete. So this particular B cell would secrete this kind of an antibody. This one would be secreting this kind of an antibody. These are Y-shaped proteins that I mentioned before that are kind of like these lobsters that circulate in the bloodstream, and they're very specific for a particular antigen. So as it turns out, even though we have tremendous diversity, and this is a strength of our immune system, it's also a little bit of a weakness, which means that it can't respond very rapidly to an infection, because only one kind of B cell is going to be able to respond to that particular foreign antigen, and the rest are sort of like spectators in the fight. But when the particular B cell becomes activated, then it'll start to dominate. And so here's an example of that. So picture like hundreds of thousands of different kinds of B cells. Okay, there's all these different kinds of B cells, dot, 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 in this direction. And they all differ by their antigen receptor. And so as it turns out, if a B cell happens to stumble upon an antigen, what it'll do is it'll become activated. And what that means is that it will simply divide and proliferate. And so just that particular kind, through mitosis, each division will be clones. And so this is known as clonal selection. So from a few cells, you get many thousands and thousands of this one particular kind. And that's really great because they'll be able to then respond to the germ up here by becoming what are called effector cells. And now effector cells are the ones that are taking care of matters, which means that they increase in size in what are known as plasma cells, and they secrete these Y-shaped proteins, which are very similar in structure to the antigen bonding protein up here, the antigen receptor. And then these things will come out and destroy the foreign cells. And that's what this video is going to be all about, and this how antibodies actually go about doing that. But I just wanted to overview the process. And now, just to continue that overview, once the germ has been defeated, the cells will remain not in the same kind of numbers that they were during the major battle. But when the war is over, it's almost like you're taking down your major troops, but you keep a few bases around in the body. So there's some memory cells that will circulate for literally years in the body. And so if that germ were ever to appear again, the B memory cell will be able to secrete a tremendous amount of antibodies very rapidly. It's known as the secondary immune response, and then you don't even get sick. And so we have these sort of, if you will, dormant memory cells that remember when there's an infection. And so what's fascinating about this thing is the way I've described it is here that the B cell happens to stumble upon an antigen. Well, the truth is that's less likely to occur because these are so few in numbers. What's the more likely scenario is that B cells are called to the scene of the crime, if you will, or the scene of the infection or the inflammation by what are chemicals called cytokines. And these cytokines, sometimes called lymphokines, these cytokines came from another type of lymphocyte called a helper T. And this helper T cell is, let me draw it in right here. This helper T is also a lymphocyte, and it's more likely that it will come into proximity to a type of white blood cell called an antigen presenting cell. The antigen presenting cell is just your ordinary macrophage. Let's say it's a neutrophil. And what happens is when a germ is destroyed through phagocytosis, it's presented on the outside of the antigen presenting cell on what's known as a class 2 major histocompatibility complex. And the T helper cell will identify that. And then this is the point that I want to make, the T helper cell will then produce chemicals. And these are called those lymphokines or chemokines. And an example of that is something called interleukin 2. It's a type of cytokine, meaning like cell movement. And it's secreted by the helper T cell. And that particular chemical, well, let me just draw it circulating right here, will then call the B cell to the area, and then that particular B cell will then become an effector cell, a plasma cell, and it'll start secreting large amounts, now that it's a plasma cell, of antibody and become an antibody secreting cell. Now, the way I described it previously was that the B cell encountered the antigen all by itself. And that's a possibility, but it's more likely to be called there by the helper cell. So let me go through that again with this diagram. So here's the germ, gets into our body. It's unfortunate. It breaks into underneath the skin. It's in our blood. A macrophage comes and engulfs it through phagocytosis. It takes some of its proteins and literally displays it on what's called a class 2 MHC marker, and it presents it. So this is an antigen presenting cell. And so as it turns out, the helper T can come along and detect that. It binds to the antigen presenting cell with the complement determinant 4 protein, and then that causes the helper T cell to become activated. What does this mean? Well, this cell produces interleukin 1, and then the binding of it causes the helper T cell to start to divide. And so when this divides, and its numbers increase, so its numbers are increasing right in here, then the helper T cell will produce interleukin 2, which are chemicals that I was showing you just a second ago that will call B cells and other T cells to the scene of the crime. Okay? And another route that can occur is that when a B cell is encountering a foreign germ, a helper T can actually bind with a B cell, and that will also class 2 MHC marker. The helper T will also secrete lymphokines, interleukin 2, which will bring more B and T cells to the area. But nevertheless, what's happening is that the B cells, and this is the punchline, when there's a germ, the B cells will become effector cells, especially the ones that can respond to it, which means they're going to produce a lot of antibodies, and then that's ultimately going to destroy the germ. So this conversation is all about how a B cell will encounter an antigen, and when it encounters an antigen, it will be stimulated and therefore divide, and it will produce a tremendous amount of antibodies that will come out and destroy it. And so usually B cells are called to the scene by an antigen-presenting cell that displays some fragments on its MHC2 marker. The helper T cell then calls the B cell using interleukin 2. But once the B cell is there, it'll start to attack the foreign antigen. And here, thought this was kind of funny. This is your typical B cell. And what the B cell is producing is a tremendous amount of these proteins per second. How about this? 2,000 antibodies per second can be pouring out one of these activated effector cells that are highly secretory. And so how does it do it? Well, the whole immune system is based on recognizing the fact that some cells are different than our own. And so all of our cells have a class 1 MHC marker, and so we end up avoiding that. But anything foreign, so it could be a germ, or it could be something that's kind of benign, like for example, like pollen, could be something that invokes an antibody response, but it's not harmful, it's not pathogenic. And so what is it about a foreign cell that makes it recognized as being different? And so it comes down to the fact that the cell membrane has proteins on it that are called antigens. And so these antigens are, quote, elicit the humoral response. And so they're typically like proteins. Some of them are proteins with some sugar on them, so they're sort of glycoproteins, the glycocalyx of the cell membrane. They're sometimes polysaccharides, but nevertheless, it's some kind of epitome, if you will. Sorry for all the different terminology, but some sort of antigen determinant protein epitome on the outside of the antigen will stimulate the response. And what is an antibody? What is this thing? Well, it's a protein, and I won't go too far into it, but this is the brief discussion of what an antibody is. It's an immunoglobulin. It's made up of, and I'll sort of highlight this. I'll go with the blue here. It's made up of one polypeptide chain, like this, and a second large polypeptide chain, like this. Okay? And so when a polypeptide chain is connected to another polypeptide chain, it often makes up what we would call a protein. And so as it turns out that there's 4 polypeptides to this one protein, there's 4 chains. And so these 2 in the center are referred to as heavy chains because they're larger. And then there's these 2 lighter ones over here. And so it's like, well, how can you have them 4 chains unless they're connected together? Well, they're connected via, if you know your protein conformations, they're connected by R groups of an amino acid called cysteine, and these form disulfide bridges. So they hold these together. But it's this region right up here that I want to emphasize. It's this region right in here between the light chain and the heavy chain, the light chain over here and the heavy chain that's variable. It is this antigen binding site that actually binds to the, quote, epitome of the germ. And this is what sort of provokes the production. Because, if you will, on the outside of the B cell, you have these sort of antigen receptor proteins that are basically the same structure as the secretory protein. So if this becomes the protein that's binding to the epitome, then the B cell will eventually become a plasma cell and secrete antibodies into the fluid, and that's what that means, plasma fluid. So this is a humoral response. And so here's an antigen, and here's these proteins or these antigenic determinants out here. And so what will happen is when the cell secretes antibody, it'll eventually attach to those antigens and either neutralize them or make them a little bit easier for phagocytosis to occur, and I'll discuss those options in a moment. Okay, so I just wanted to mention the fact that this is the epitome right here, and this is the antigen binding site right there of the antibody. And so here again is a more detailed look at this. And so here are the two heavy chains, because they're larger, the two lighter chains. Here are the disulfide bridges holding the whole structure together. So it really does look like a letter Y. It really does, or like a lobster. And up here is the claw. And this is where it's different. We have hundreds of thousands of different kinds of antibodies based on this variable region right in here. So this variable region, this V area, it's how it's different. And it has to be different because each antigen has a varying epitome. And that's how the antibody is able to bond with the foreign organism by that particular area right there. And so, as it turns out, in the lab, if we're able to isolate a particular B cell and grow it in culture. So in other words, you have your petri dish, and you're isolating your B cells, and they secrete a particular type of antibody. We can then culture, maybe in a liquid, we can culture those B cells and have them produce a tremendous amount of that particular kind of antibody called monoclonal antibody, because it comes from the same B cell and it's very specific to a particular antigen. And so that's useful, because then we could take, as a form of medicine, we could use these antibodies against particular germs. And for example, an example of that would be, this is kind of the latest thing, if you will, although it depends on, that's relative. But I find it kind of recent in terms of discovery, is that we're at a point where if a cell in the body becomes a tumor, and so it's a cancerous cell, and it has a particular epitome about it that can be identified, what we can do is, in culture, grow these antibodies. And if the antibody attaches like this to this tumor cell right here, what we could do is connect, how about this? We can connect like perhaps a radioactive isotope or some sort of lethal chemical or some sort of toxin-linked antibody is what this is called. And it would specifically search out like a tumor cell. This would be a tumor cell down here, tumor cells. And so the antibody searches amongst the whole body and it finds these tumor cells, and then this provides like a lethal dosage of radiation, and it destroys the tumor specifically. So there's a lot of cool things having to do with monoclonal antibodies. In general, I mentioned there's hundreds of thousands of different ones, and they all differ by that variable group right at the end of the molecule, if you will. Here's the light chains like this, and so it's this area right here that's very variable. But I wanted to point out that there's 5 kind of major classes of antibodies based on sort of their heavy chain and whether or not they're single or they work in groups. And so there's immunoglobulin M, and it's a pentamer, meaning there's 5 of them that work together like that. That's kind of interesting. There's immunoglobulin G, which is a monomer, and then there's a dimer, which there's 2 of them, immunoglobulin A, immunoglobulin D, and immunoglobulin E. And now there's a variety of these 5 types, that I just wanted to point those out. So if you're ever studying antibodies in particular, you'll certainly be familiar with the fact that there's these 5 basic kinds. So the final part of this conversation I want to talk to you about is how antibodies actually go about rendering and destroying antigens in other words, like making them harmless or actually just destroying them outright. And so the two basic mechanisms of destroying is getting a foreign cell to bust open or to lyse, or making a foreign cell easy to be destroyed through phagocytosis. So these are the two ways. Okay, either the antibody makes it easier for phagocytosis, or the antibody causes the foreign cell to bust open. Which is called cell lysis. So let's look at the detail of that. So we have something called neutralization, and if an antibody attaches onto a virus or a bacterium, what it can do is actually attach to the binding site that coats the bacteria, and so it blocks that, and so it then makes it useless. It doesn't. It's not harmful anymore. And in some instances, agglutination can occur, which causes the bacteria to clump together. When one of the cool things about the antibody is that it has all these different attachment points, and so you can get foreign cells. You might be familiar with antibodies are used to type blood this way, and so antibodies cause red blood cells to clump or agglutinate, and that's how you can tell what blood type you are. And so if the cells become clumped, it makes it easier for a macrophage to come along and take care of them. And then likewise, sometimes it can take these foreign proteins or foreign antigens and clump them together like this and make them come out of solution, something called make them less soluble or precipitate. And so therefore it makes it a little bit easier for white blood cells to destroy them. And then finally, I'll get into this in a little bit detail in a moment, but antibodies can attach on the surface of a foreign cell, and there's circulating proteins in the plasma called complement, and then the complement attaches to the antibody, and then it jumps off and forms these little pores, which then leads to the cell lysis. So let me have you look at some of these in more detail. So neutralization, I mentioned, is when the antibody blocks the activity of the antigen. And so what that means is the antibody literally attaches to the molecule that the virus uses to infect a host cell. And so if the virus is using some kind of protein to attach to a cell to invade it, what the antibody does is blocks that from happening by attaching to the antigen, therefore making it the activity less. Okay, and so sometimes what will happen is a process called optimization. Optimization is when the antibody helps to enhance the macrophage's ability to attach to the germ. So if here's a germ like this, and it's like that, and you're having these antibodies attached to it, it's sometimes easier for the macrophage to get a hold of the germ when there's antibodies attached onto the outside of it. So it's kind of similar. Both of them are involved in phagocytosis ultimately, because if it's not binding to a cell and invading it because of the antibody, then the white blood cell can engulf it. And then this is making it a little bit easier, so it's enhancing the macrophage's ability to phagocytize it. And then I mentioned agglutination, is that agglutination makes it possible when, especially with immunoglobulin M, when it's that pentamer, when you have many bacteria and viruses all sort of clumping together. It's sort of like throwing a net over like a bunch of criminals, and you can like gather them all together, and then that makes it easier for other monocytes or neutrophil to come and phagocytize them. And then finally, precipitation is when you get some of these like cross-linking between antigens, which then causes them to precipitate, because the antibodies are attaching to them, and therefore it makes it easier to be destroyed. And then finally, this is actually not to play favorites, but this is my favorite. This complement fixation is totally remarkable. Well, you've got, here's the cell membrane of a germ. Okay, Okay, if you're picturing this, here's the foreign antigen or the epitope, and here's an antibody. And when an antibody attaches to it, and you're like, well, what's it doing? It's like, well, it attaches to it, and then in the plasma, which is up here in the plasma, you get these circulating, that means like flowing proteins. They're plasma proteins called complement. And when enough complement attaches to the heavy chain of an antibody, what will happen is that it'll cause these complementary proteins to jump off the antibody. And they're called membrane attack complex. I love that name because they come and they actually form a pore that will occur in the bacterial membrane, which will allow like solutes to come in or water to come in and cause the cell to lyse. And it's incredible. And so it's kind of it begins often with the immunoglobulin M or immunoglobulin G, and it binds to the pathogen. And so then what happens is that these complementary proteins again accumulate on those antibodies and forming the MAC or the membrane attack complex, and that ultimately results in the lysis of the cell. And so if you're, again, finally, you might have it by now, but this is, the membrane of a bacteria. This is the germ. And so, for one thing, you've got these circulating antibodies, okay? And these are cruising around, and then here's the surface right here, proteins of the germ. And so you got the antibodies attaching to it. That's one thing. And once they attach to it, then you have these complementary proteins which are attaching on the outside. And then once enough complement occurs, then these jump off and form like a membrane attack complex, which then allows for the movement of fluids, and the fluids into the cell, which then cause the cell ultimately to bust open and be destroyed. So that's pretty awesome. So I hope you enjoyed, remarkably, and you may be surprised, but a brief look at the humoral response of our immune system and the importance of B cells and how they secrete immunoglobulins known as antibodies. Thanks for watching.